Hey guys, my name is Z and you're watching MakesItEasy. And welcome to IGCSE Biology edition where topic 1 we have nutrition. And for the topics in nutrition that I'll cover are photosynthesis, factors affecting the rate of photosynthesis, plant minerals, adaptations of the leaf, balanced diet, energy content of food, the digestive system, enzymes, acids and biles, and the adaptations of small intestine. So first we have photosynthesis, and photosynthesis is carried out by plants in chloroplasts, and the equation for it is carbon dioxide plus water equals glucose plus oxygen. And besides photosynthesis in the day, the plants also respire all day. And here's a glucose store, and glucose is carbohydrate monomer, and we'll look into it later here. And as a glucose, the glucose stored in animal, bacteria and fungi are glycogen. In plants, it's starch, and in fruits specifically, it's sucrose. And glycogen, starch, and sucrose are all insoluble. And here's a range of polymers: lipids, which are fat, carbohydrate, and protein. And the monomer for lipid are glycerol and fatty acid. The monomer for carbohydrate is glucose, and the monomer for protein is amino acid. And the main elements found within the monomer, they all have carbon, hydrogen and oxygen. Except for protein, it also has nitrogen. So here's different test measurements. For quantitative versus qualitative, the quantitative test is information about quantity as the name suggests. And qualitative test is information that is descriptive, such as language and regards to phenomena which can be observed but not measured. And here's accurate versus reliable versus precise, which you can read. It's a bit complicated and confusing, but here's a more visual diagram. So reliable, it's like how consistent, but not valid. And both reliable and valid are it's consistent and it's on target. The result is what you get from your theory work. So here's polymers, more on polymers that we talked about just now. We have lipids, the monomer is glycerol and fatty acid, and glycerol is C3H8O3, and it's basically alkane with 3 hydroxyls, and fatty acid is an alkane. So here's glycerol and alkane for fatty acid. It's carbon atoms with hydrogen atoms along the length of the chain. So glucose for the monomer of carbohydrate, and its basic units of carbohydrates are simple sugars such as glucose in cells and fructose in fruits. And amino acids are the monomer for protein, and there are only about 20 different naturally occurring amino acids, but each protein molecule has hundreds or even thousands of them joined together in a unique sequence, which happens in the ribosome in the cell. So here's more on biological molecule recap for year 9. Enzymes end with an ASE, like lipases, lipase, protease, and carbohydrates. And assimilation is making polymers from broken down monomers, and digestion is breaking down polymer and turning it into monomer. So here you can just look at the purpose of each polymer here. So moving on, we have biochemical tests, and it's basically tests to test those biochemical stuff, like polymer and stuff. So here's glucose. The test for it is Benedict test. And there's a color and how do you do the test. For starch, it's iodine test. Protein, it's a burette test. And for fat, it's emulsion test. So moving on, we have factors affecting the rate of photosynthesis. And here we have light intensity. And we have a curve over here for light intensity and the rate of photosynthesis. And you can have a read for the description for part 1 and part 2, respectively. And here we have carbon dioxide concentration over here on the x-axis, and the y-axis is the rate of photosynthesis. And similar to this one, you can read the part 1 and part 2 respectively over here. And the key thing to write or to note that is you have to know this concept of no longer limiting. It's no longer a limiting factor. And then we have temperature and humidity. As the same, we have temperature and rate of photosynthesis, and here's a description for it, as well as humidity. And you can pause if you want to have a read. We'll move on now. So here are plant minerals. I'll just focus on the function and you can have a read for how it's obtained and the deficiency. And for calcium, calcium is used to make the calcium pectate, the glue that sticks adjacent cell walls together. And for magnesium, it can be found in the enzymes of the chlorophyll in plants and it's used to make chlorophyll. And plants use nitrates as a supply of nitrogen since nitrogen can't be directly absorbed by plants, and it can be absorbed by plants using the nitrogen cycle in year 9, which you'll look into in the next slide. And phosphorus is a vital plant nutrient and its main use of their phosphate compound is in the production of fertilizers. So you can have a read for the deficiency and how it's obtained by yourself. So we'll move on now. The nitrogen cycle. So nitrogen can turn into soil nitrates from 2 processes, via lightning or via nitrogen fixation by nitrogen fixing bacteria. So soil nitrates, then it's a cycle, and soil nitrates can turn into plant protein, to plant protein, or from the denitrification into nitrogen. So it can go into 2 ways. You can see different things. And then we have plant minerals and plant cell. The cell wall holds the plant's shape. The cell membrane controls what gets in and out the cell. The permanent vacuole stores water and other nutrients. The nucleus contains the plant's DNA, and for the chloroplast, it contains chlorophyll, which is used for photosynthesis. And mitochondria, aerobic respiration happens in the mitochondria and it releases ATP energy for us to survive and for us to respire. And cytoplasm is where chemical reaction takes place, and ribosome is where proteins are made from mRNA and tRNA and other stuff. So here's a cross-section diagram. So moving on, we have adaptations of the leaf. So we have different types of adaptation. We have large surface area, waxy cuticle, green contains chloroplast, upper epidermis is transparent, has a stomata, it contains veins and it's thin. So you can just read the different description. And for the leaf structure, there's a waxy cuticle on the top and it's waterproof. The upper epidermis, then the palisade layer, which contains the palisade cells over here, and the spongy layer over here, which contains air spaces to allow carbon dioxide to diffuse through the leaf, and the lower epidermis, which contains guard cells that surround an opening called the stoma or the stomata. So here's balanced diet, which is excretion and ingestion and nutrients. So excretion is like pee and sweat, where it's excretion of the waste products of metabolic reactions. And ingestion is like feces, which is an expulsion of undigested food, and it's mostly cellulose. And you can see respiration versus breathing. And for nutrients, there are generally 3 types: macronutrients, micronutrients, and others. And macronutrients include carbohydrate, proteins, and lipids, which are polymers, and micronutrients, which are vitamins and minerals. And others include water and fiber. And here's macronutrients in our diet, where you can have a read. And moving on, we have micronutrients, which are vitamins over here, and minerals over here, which includes iron and calcium, as these 2 are one of the most important minerals in the body. And then we have others, which include water and fiber. So we'll move on in a few seconds. So moving on, we have energy content of food, and energy transfer is equals to mass of water times 4.2 times the temperature increase, and energy transfer is in joules. And for CORMS, it's an acronym that stands for change, organisms, repeat, measure, and same. And CORMS is an acronym for how to design an experiment, and here's the meaning for each acronym. CORMMS. And here we have an energy content of food experiment, where we investigate the energy content of food. So here we have the CORMS and the CORMS 6-mark paragraph, and we have to turn all of these into a paragraph over here. And here's a step-by-step plan if you decide to do it. So here we have the digestive system. We have the mouth, the salivary glands, esophagus, and the stomach, and the small intestine and large intestine and stuff. and here's a more simplified version. Then we have the digestive system function, and these are quite in-depth, and it's quite complicated for IGCSE. So don't worry if you don't know any terms or any a name or any definitions for the thing, because it's quite complicated. You just have to know the main ideas and the things that are in IGCSE, like teeth, liver, and salivary glands and stuff. So next slide, we have another different names and the function. And then we have monomers and polymers. And monomers are single simple molecules or compound, and polymers are a chain of repeating monomers chemically bonded together. And the nutrient is a substance which is needed for growth, repair, and metabolism. And the three main nutrients are carbohydrates, proteins, and lipids, which are polymers. So from a monomer to a polymer, it involves a process called polymerization. And here are the three main nutrients, or the polymers. Then we have biological molecules recap from the first few slides, as we talked about here just now. And it's meant to be a typo. It's meant to be where the enzyme to break down the polymers are made, which we'll focus on the next slide. So from here we have the enzyme. So the food group protein, the enzyme that digests it is protease, and it's made in stomach, pancreas, and small intestine. For carbohydrate, it's carbohydrates, and it's made in saliva or mouth, pancreas, and small intestine. And lipase, which is the enzyme that digests lipids, are made in small intestine. So you can see here. So what it's broken down into is basically the monomers. And then we have acid, enzymes, and biles. So you can have a read for yourself. On this side here, on the left hand side, we have the stomach acid. And on the middle, we have enzymes. And on the top, on the right, we have bile. And moving on, we have adaptations of small intestine, which is digestive system recap. Number one, we have esophagus over here, which is a tube that connects mouth to the stomach. Number two is small intestine, and it's carbohydrates, proteins, and lipase enzymes from pancreas digest food groups, and it is absorbed into the bloodstream here. And large intestine is where water from food and secretions is absorbed into the bloodstream, stomach, and mouth. And then we have the villus, and we have the adaptations of small intestine, and the villus is located in the small intestine in the lining. And the walls are only one cell thick to reduce diffusion distance, so it increases diffusion rates for absorption to happen. And it has an extensive network of blood capillaries to transport glucose and amino acids away from the small intestines and through the blood, so it maintains a steep concentration gradient. And it has veins that transport blood away from the small intestine, an artery that transports blood into the small intestine. And lacteal has which is an internal structure called lacteal, which transports fatty acid and glycerol away from the small intestine. And for large surface area, it contains microvilli on the surface of the villi to absorb more nutrients with larger surface area. And lastly, we have a cross section and location of the villus. So here's the small intestine. We have a network of capillaries over here, veins and arteries. And then inside the lining, we have villus or villi, like here, small intestine here. And zooming into this, we have the villi, network of capillaries. And then zooming into the villi, we have microvilli. So these are the intestine, villi, and epithelium. So that's it for this IGCSE Biology series for nutrition. I hope you guys have enjoyed it, and drop a comment if you want me to do more videos like this. But I'll see you guys in the next video. Until then, stay safe and happy learning.